here as you're driving a cab in New York City that should be kept in a cab. It's just like going to Vegas. As for Madison Square Garden, yes, I was the fastest flipper, and I had no choice because when games are over, Knicks games or Rangers game, any of the games that are play at Madison Square Garden, you get a deluge of people, thousands of people who are really, really hungry and they're waiting for a pizza on their way to a bar. So we have no choice but to get good at making pizza and making them really fast. So what I want to do is just give you a quick overview of who I am, how I got in here, and then I'm going to tell you a little bit about my work, and then at the end of it we can come back and talk about my experiences. And I think as we do this, it will be, I think, very helpful to just make this very informal, ask questions, comments, anything you like as we go along. So I'm originally from the Dominican Republic. I was born there, and then I moved to New York City, to the Bronx, when I was pretty young. I went to high school in the Bronx, James Monroe High School, which is not a high school anymore. The building is modified to be something else nowadays, as I hear. And then I stayed in the Bronx, and I went to Lehman College, which is part of the City University of New York. Do you guys have a pointer, by the way? No? Well, while you're checking, so when I was in high school, I wanted to always keep myself busy, keep myself out of trouble. Also, I wanted to have the latest Jordan kicks, so I worked at different places to make some extra money, and I did a few other things in high school as well. I was part of the track team. I used to be fast, long time ago, and I also did this part-time after school work. I got paid pretty well for doing minimal work. And so then, as I say, I went to Lehman College, which is part of the City University of New York, and that college is in the Bronx. It's actually right on the border between the Bronx and Yonkers. And I majored in biochemistry. As I was majoring in biochemistry, I met an awesome guy who was actually happened to be a graduate student who was teaching one of the courses that I took, and he introduced me to his research, and he asked me if I was interested in doing any research, and I jumped at the opportunity, and he introduced me to his mentor, his PI. I joined the lab, and a few months later I was awarded an MBRS fellowship, a minority fellowship, to do research in that lab, which I did for 3 years. And so I worked in plant genetics, doing carotenoid, corn type of research for all those 3 years. And when I was doing that, just to give you some perspective of how you choose to do research, I chose to do that research because I had no choice. It was an opportunity that I just couldn't pass on. But then I found out that actually it was an amazing kind of work that not only was that lab trying to get done, but other labs around the world. And the purpose of it was to try to make rice make vitamin A compounds. So rice do not express compounds that can be turned by our body into vitamin A. And this is really important. It's important because most of the population in the world, India and other places, their main diet is rice. And so they lack vitamin A, and so they tend to suffer from vision issues and just general developmental issues where vitamin A is really, really important. So we're trying to figure out how vitamin A, or the compounds that are the precursors to vitamin A, are produced in corn, so that then we can take that information and modify rice so that they can make vitamin A, genetically modified rice, so they can make vitamin A. As I was doing this work at Lehman College in plant genetics, I got a bug of wanting to know a little bit more about my system, the mammalian system, and so I joined a lab. I obtained a fellowship to do a summer internship at Einstein College of Medicine, which is also in the Bronx, and I worked there for 3 months doing research, and the aim of that research was to try to make bone marrow cells better so that they can be used for bone marrow transplants. And so we're doing a lot of genetic molecular biology to try to modify those bone marrow cells. When I finished, I had a lot of, when I finished college, I had a lot of skills, just basic molecular biology, genetic skills. And so I was hired as a lab technician at Stony Brook, and I worked there for 3 years, and that was in a neuroscience lab, did some neuroscience work, and that's when I truly got the bug about neuroscience and to keep working on the mammalian system and shift from plant genetics. And that actually was, I would say, was a really, so some people were questioning my decision to work, and rather than going straight into graduate school. And actually, I really loved that I took that time in between college and graduate school. Everyone is made a little bit differently, but I felt that I needed a bit of extra time to just recoup from all the work that I had done on my own, pretty much, during college. And so I took advantage of my found wealth to travel around the world a little bit, to get to know places that my mother and my family couldn't afford for me to see and to be exposed to. After I was done with all of that, I applied for graduate school at Stony Brook. I was accepted, and I stayed in the neuroscience department where I did my PhD in neurobiology and behavior. And I think I had a very, very productive graduate school career. But, graduate school and undergraduate is as good as the mentors that you have and people that you have, but also as how much you put into it and the vision that you have for yourself. And so as I was making these transitions through high school, college, and graduate school, at each step I had really awesome mentors that were helping me. And I was, obviously doing my part to be accommodating so that their help was a little bit easier and they could help me a little bit more reach a lot of the goals that I was beginning to set for myself. When I was in graduate school, I realized that there's different ways to have goals. You can have general goals of what you want to do, and one the simple thing is I want to be successful. Right? And then you can dig down a little bit more. I want to be a successful medical doctor. I want to be a successful researcher. I knew at this point that I wanted to be a successful researcher, but then you need to have a more detailed plan on how you're going to accomplish that. And in my very early years as a graduate student, I realized that I needed that detailed plan. And so I started to think about what area of neuroscience, biology is incredibly vast. What area, specific area of neuroscience was of most interest to me, and how do I continue to make progress so that at some point I can become one of the experts in that field of neuroscience and therefore be very competitive with anyone around the world in this kind of research. And so that started early on in graduate school. I dove into neuroscience and looking at axons and looking how the nervous system is put together. And then I moved up onto Harvard, where I continued some of that work, but at a more system-based level. I was working with molecules and cells, and I started to work with animals while I was at Harvard, so that I can add to my repertoire of skills and knowledge base and continue the career path that I had set myself for. And while I was there, again, I was helped by incredible mentors, not just the PI of the lab, but also graduate students, postdocs, technicians. I mean, everyone that is around you in some ways is a mentor if you are open to what they have to teach you, and also sometimes just their ideas. I was also helped by, along all this way, by minority fellowships. I was at Harvard. We had a 2-year supplement to the grant, and it helped pay for my salary. It helped pay for my research. I obtained quite a few fellowships, travel fellowships that allowed me to attend national meetings. One of them was in a Keystone Symposium in Lake Tahoe, really nice place. Another one was in Connecticut. So, support extends throughout, and it comes in many, many facets, many ways. But as I was setting myself and settling down on the knowledge base and the skills that I had acquired, had also developed this vast network of mentors and friends in the biology, in the field of biology. I started to give back a little bit more. I went back to Lehman College to give a couple of talks and meet with students just like we're doing today. I joined the board of Tutor. It's a mentoring program at Harvard to mentor undergraduate students who want to do research and who are doing research. So I was their outside mentor to make sure that they're PI were not taking advantage of them. They were on the right path, and someone just to bounce off ideas of. And so there I mentor about 10 students, 8 to 10 students. And then I had another opportunity. I'm a member of the Society for Neuroscience. It's a big meeting, over 30,000 people from all over the world attend this meeting. But there is a lack of minority representation with the Society for Neuroscience and throughout the neurosciences, as a matter of fact. And so I took advantage of a program that they had to mentor a graduate student who was a graduate student at Harvard, Stanford at that time, a minority graduate student, to mentor him through his graduate course. At that point, I was in my 4th year as a postdoctoral fellow, so I had a lot of skills that I thought were, and experiences that would be very, very beneficial to him. So then after Harvard, I came here. Actually, I didn't come here to Blacksburg. I was hired to join the new budding Research Institute and Medical School in Roanoke, which is the top building, and it's actually the second building here, over here. It's actually, I had no idea about Roanoke or Blacksburg, had never been here, so I was quite apprehensive growing up in the Bronx and always living in cities and coming to a place like this. But when I flew into Roanoke in that little propeller plane, which was still kind of scary, I thought I was going someplace not so great. I realized and I saw how beautiful the place is. There's actually quite a bit of diversity here. There's a lot of energy, a lot of young people here, which is great. So I was taken by a lot of different things, but certainly at the Institute we were putting together an incredible team of researchers. I'm one of 22 now, with 2 more likely coming on board that are coming from many different institutions, Baylor and you name it, many other institutions, Harvard. So great place to be, great place to live. And with me, I brought my kids and my wife. So the little one on the left is Benji, the bigger one is Nikki. And they had to come with me, not just because they're my kids, but they're also tough. And anyone messes with me, and they're going to hear it from this guy. So they're, it's kind of funny. I left the Bronx, but I guess my Bronx attitude hasn't left me. It certainly has percolated my kids, so, which I think is a good thing if you use it right. So what I like to do is come back to the first slide that I just showed you towards the end, and just open to questions and for us to have a conversation. What I want to do now is just give you a very brief overview of what I do, just general stuff. Okay, so as Nina mentioned, what my lab is doing is studying how the nervous system, but also muscles, change with aging, and how they're affected by a variety of diseases, from muscular dystrophy to Lou Gehrig's disease, or ALS, amyotrophic lateral sclerosis, Alzheimer's disease. With aging, just normal aging without any diseases, we lose motor skills. Our motor skills deteriorate, they erode, and also cognitive skills. You can see this in athletes, with motor skills, for example. Most of these athletes, amazing, probably not so much anymore, especially these guys, they're slowing down, so it's amazing, some of these athletes, they peak, and depending on the sport, they peak by 26 years of age, and then, they just can't compete. That's more of a difference, milliseconds of a difference. It's a matter of whether they can be in the Olympics or not. The same thing with the swimmer here. LeBron has probably another 6 years of being, he's 28 now, another 6 years of being super competitive, best player in the NBA, but after that his motor skills will really begin to leave him. And so just like it's happening to Kobe now, well, he's doing really well this year. So this is going to happen, and those guys are young, 34 years of age, it's not an old person. But at that young age, the motor system is going to begin to erode. And, eventually it's going to happen. It's going to happen to them, it's happening to me, it's going to happen to all of us. It's just a matter, fact of life. And with this, clear hourly physiological changes, so these are physical changes that you can see in anyone, right? You saw in my kid. My kid, the youngest one, he looks like almost like a baby, the 5-year-old. That's a little bit different. I can see different faces here, different ages. And you can see that with Arnold, who certainly is not the Terminator anymore. He's actually, he terminates something else now, but no, other weights. when you look deeper into the system, into the body, you see clear changes that happen as as we age. The brain atrophy, there's a reduction in brain mass, but also the circuitry, all the circuitries from the hippocampus to the cerebellum, all the circuitries that we utilize and we don't need to think about it because this is what makes us think and move. Those areas of the brain begin to deteriorate slowly. And so what I'm doing to get at how this happens, we know hourly, when we look at a person, when we look at a structure, a brain or a muscle, we know there are clear changes. But how do those changes happen, and why do they happen? There are molecules, there are lifestyle factors that influence how we age. And so what I'm doing is taking advantage of this species here. I'm using it as a model system to study how the nervous system and muscles change with aging. And for a couple of simple reasons. One is I can't do this kind of work with humans. I can't just ask someone, donate your muscle before you pass away, or at the prime of your life. But also it just allows us for a lot of manipulations, genetic manipulations. We can isolate cells. We can do a lot of things with model systems. And these animals, they live for 3 years. These are mice. So mice live for about 3 years. And just to give you some perspective, a 2-year-old mouse is more or less equivalent of a 75-year-old person. So we can do this kind of research if we're studying aging much, much faster. I can do it before I die, actually. So I use this kind of model system to look at this. Right? These are Purkinje cells in the cerebellum. Here are other Purkinje cells, and these are the apical dendrites. These are the connections. We're making connections right over here with other neurons. And so this is from a mouse. It's from a mouse that was genetically engineered to express different forms of a fluorescence protein. And so we can see them, and we can see them individually and where they're going and then how they connect. And so that's the kind of work that we're doing in the lab in the brain. Oops. We're also doing other work out in the periphery. So with us, as with mice, we have spinal cord that are populated with motor neurons that send axons out to the periphery where our skeletal muscles are. They make contact with skeletal muscles, and this is a synapse. This peripheral synapse is called a neuromuscular junction. So we're populated with synapses in the brain, in the spinal cord, and then in the periphery we have these synapses which are called neuromuscular junction. This is the final output of the nervous system. When you think of when I think about moving my jaw or finger, information goes through my brain to the cerebellum, down the brain stem, to the spine, to the motor neurons in the spinal cord here, and then they elicit a muscle contraction so that I can move my a finger or my leg. This is the final output. We lose this connection and we're done. And so in a disease such as amyotrophic lateral sclerosis, Lou Gehrig's disease, the first thing that happens is that this connection is lost. The neuron retracts, dies, and then unfortunately we can't move. Eventually we can't breathe because our diaphragm, which is controlled by this kind of connection can contract. So very, very important. So I used mice, as I showed you before, to look at muscles and look at this connection. These are 2 skeletal muscles from a mouse. These are just beautiful little images, as you'll see. This is the site on the muscle fiber. So these are 2 individual muscle fibers that, imagine any muscle, your tricep. They're one of hundreds and sometimes thousands of individual muscle fibers. And they have these receptors right on the membrane. It's like a docking site for the nerve to come and make contact. So these are nerves that are coming to those receptors, paralyzes us, and that's how it actually wins out. And this is the axon. These are axons that are coming to make a connection, the green color, and that's using this genetically engineered mice that allows us to visualize neurons in the central nervous system, but also out in the peripheral nervous system.
So you're interested in everything we see, or just the...
Yes. Yes. So, yeah, good question. So, in biology, everything just, in some ways, there's a lot of similarities as when you look a little bit closer with anything, right? It's just like, with career development, you need a team. You need a lot of people to help you really, to succeed. Here, in order for this connection to function properly, the muscle has to be healthy, the neuron has to be healthy, the axon has to be together. So in some ways, I try to look at the micro environment and sometimes the macro environment to see how it affects this small structure within the muscle. Okay. So one of the things that I've done and that we do is we ask a very simple question: how does lifestyle factors, whether it's diet or exercise, affect synapses? And so I take advantage of that synapse that is much easier to see, visualize, work with, and central synapses to see how some of these lifestyle factors affect synapses in general. One of them is caloric restriction. You guys already ate, so you feel a little bit guilty, but not so much. Here is a young synapse. Here is the muscle end, the receptors. Here's the nerve, visualized slightly differently. And this is just an overlap between the nerve and the muscle end. Here's an old synapse. This is from a 6-month-old animal mouse. And this is from a 2-year-old mouse, and you can see clear differences between the two. This one has broken down into little pieces. The nerve is not quite overlapping with the muscle, with the receptors. So that bridge of communication is broken. And when you have a broken bridge, you can't really, you can't send information, you can't traffic things back and forth at all sometimes, or effectively. And this is what happens when we look at an old mouse, same age as this one, but this is an old mouse that was on a calorically restricted diet. Now that synapse looks like the young one. Okay? So what this told us is that this lifestyle paradigm can slow synaptic or protect synapses from the ravages of aging. Here's another lifestyle that we all try to pay close attention to, but someone like me, sometimes we just are too lazy to hit the gym. And this is exercise. So this is a mouse that is already old. I put it in a cage with a running wheel and allow it to exercise for a month, and then look at the synapses again to see how exercise affects those synapses. Here's an old control mouse that was not exercised, and here's a synapse from an exercise mouse. As you can see, these two look quite distinct. Topologically, architecturally, this looks like a young synapse compared to this one. So exercise, like caloric restriction, the two best, as far as we know, anti-aging factors can slow aging of synapses. So this is kind of a proof of principle that a lot of things that we know that we avoid and we don't do very well actually do have beneficial effects, at least in mice, and we do know that they have a lot of beneficial effects in human from epidemiological type of studies, looking at human with, that exercise more, that exercise less, that are wheelchair bound, and so forth. So in my lab, what we're doing is we're just to kind of give you a brief general summary, what we're doing is taking advantage of some of the things that I did as a postdoctoral fellow to look at how neuronal and aging affects neuronal and muscle function, and what are the things that begin to go away, what are the building blocks and molecules that begin to go away with aging. Because we know that there are detrimental changes that happen to the synapses, but in general to neuronal circuits. We know that, as I showed you, the diet and exercise, just simple lifestyle paradigms, can slow aging of synapses. So we can take advantage of this type of interventions to ask what are the molecular changes that are now different in an age exercise synapse or caloric restricted synapse versus a control synapse. There has to be differences. And so now we can take advantage of this to probe those molecular factors. And so what we do to do that is we use genetic, biochemical, molecular, cellular approaches. We use kind of a combination of a variety of different tools. Do a lot of surgery in the lab. You were asking, do I look at everything? We do a lot of nerve injury, nerve cutting, crushing, transections to look at how the nerve regenerates in a young animal versus an old animal. And then we manipulate the molecular milieu to find molecules that are responsible for promoting nerve regeneration. And so what I've tried to do in my lab is just to establish, and I think as you go through different phases of your career, you want to be part of this, and if it's not there, you want to kind of try to establish. You want to be in a dynamic and a collaborative environment. Everything is too complicated. everything is too hard, and so you don't want to be in a place, nor do you want to sink into into a mental emotional situation where it seems like everything is too hard or too difficult to accomplish. You certainly don't want to do everything on your own because it's too difficult. Everyone has different strengths, different ways of looking at different problems. So you want to be as collaborative as possible, and in the immediate vicinity, whether it's in the lab that you're in, with your colleagues here, but also seek other people that can help you with whatever it is that you're trying to get at. And so, in my lab, I'm trying to instill is for everyone to be very open about what they're doing. We have lab meetings, we talk regularly. We also have joint lab meeting with another lab that does similar work, slightly different, but similar enough so that we can bounce ideas off of each other without any barriers. And we have people that come to the lab and get to listen to our research and give us feedback. And with a postdoc, and I have an undergraduate from here from Virginia Tech, I have a postdoc, technicians, and medical students. I'm encouraging all of them to participate in a meeting and also to go to local meetings, but also national meetings, because again, it's really important. As a member of MBR, a minority fellowship that I had during my undergraduate years, I was able to attend a variety of meetings in Atlanta, in California, and boy, I got to meet a lot of really interesting people that just, maybe because they were in a different location of the country, so science and career paths slightly different than my mentors, but they were all just as helpful. So what I like to do is just come back to this very first slide, and we can talk about my research if you want for a little bit, but I just like to have a conversation with you guys about anything that I've done and how some of the difficulties, because everyone faces lots of them, that I had as an undergraduate and graduate student.
That's actually what I was going to ask you. Did you experience any challenges through your path, and would you mind sharing some and how you overcame them?
Yeah, I faced. Again, everyone experience is going to be slightly different. So, how the challenges that I face are going to be different than the challenges that you face or are facing now. One of the first challenge that I faced was English was not my first language, and I was actually the first, probably maybe like some of you here, the first person to graduate from college, with 10 and then graduate from college. So I actually had to put myself in college, have to find out how to do something that now I find pretty simple, which is to fill out a college application, send it out, and then wait for the response. So those were some of the simple challenges, and then the language barrier, the cultural differences that I had to overcome. Everyone gets very comfortable in their environment. I'm certainly very comfortable in my Dominican/Latino environment, so I had to get out of my mold a little, and I did. And that actually was a great thing. It really opened up my mind to a lot of other possibilities. So that was the first challenge that I faced, but that was mostly in high school, some of it in college. In science specifically, the first challenge that I face is that science is not, it's not looked at least for where I come from, it's not looked at as it's not a very attractive career path. It doesn't have the allure that being a business person or a musician has, because it takes a long time to make money. It's a lot of work. And so I think maybe 30 years ago in science was, if you were a scientist, people had a lot more respect, and that carried the day. But nowadays, at least when I was in college, people didn't look at science as I thought people used to, I think people used to. And certainly not in the community, in the environment that I was at. The pressure was to make money. How soon will you be able to get a real job after college, and how much money it will pay. And so I would think, to me that was the biggest challenge, because it's something that you hear all of the time. And this comes from a lot of people that I respect greatly, I still respect them greatly. But if you take a step back, you realize that although they want you to make money as fast as you can, they try the same thing and they're not doing so great. So something is wrong with their advice, right? Something was wrong with their advice, and I wasn't sure exactly what it was, but I wasn't buying the advice that they were giving me, which is, why don't you just go do something else? Why are you working so hard? So that was the main one, because it's it's a challenge that affects your day-to-day life and your acceptance in in your niche, right? In your community, which is really your nucleus, your home, your family, and your friends. So, yeah.
What was the difference in the diet and exercise you did for the mice that their synapses is improved?
So the difference is, with the diet, the mice the mice were on a lifelong caloric restricted diet. So they ate 40% less calories than normal mice do, and with exercise, the mice were already old, and they were allowed to exercise for 1 month. So that is the biggest difference. We looked at mice that were in a diet paradigm for pretty much the whole life versus exercise, which was a very temporary intervention. Yeah.
Okay, I have a couple of questions and a comment. Yeah, that's right.
Only a couple?
Yeah, so going off of the restrictive diet experiments, so did you do any experiments to actually look and see if the restrictive diet was life stage dependent? Meaning that, if you're developing as a young person, you can eat whatever you want, and then all of a sudden you hit a certain age when then having a restrictive caloric intake is beneficial or not?
Yeah, that's an excellent question. So, I'm leaving out a lot of details. But in fact, when I say lifelong, the mice were put on a caloric restricted diet beginning at 16 weeks of age. So they had passed that critical stage of development. I certainly don't want to put my kids on any diet right now. I want to do as much as possible.
Exactly.
And so yeah, that's a very good point. So with this kind of experiment, you do want those critical period to pass. You want kind of puberty. Also to pass, and then once the system has settled, then you want to see if you can keep it at that optimal, right, metabolic, immunological state that you have. That I'm losing that. You're probably younger than me. So that's what we're doing, early adult. And so a mouse that is 16 weeks is what, 4-month-old, 3-month-old mouse, 4-month-old mouse. That's an adult mouse.
And the second question goes along, using toxins to induce paralysis.
Mm-hmm.
Now, is that analogous to actually neural degradation that is seen with these types of diseases? Because I know, depending on the type of venom you use, it could affect the actual neuron in different ways. It doesn't just cause the neuron to die. Molecularly, there are different pathways that I guess are affected. So when you look at those sort of pathways, is it the same as the disease pathway that's being affected?
Yeah, so let me clarify a couple of things. So the toxin what I showed you was kind of, those are sacrifice. Those are tissues from a sacrificed animal. So the tissue is dead for the practical purposes. So we're using the toxin not so much to interrogate the cellular functional biochemical changes. We're using the toxin just to be able to label, right, to see the structure, and that's all we're doing. Now we do use this toxin to label the structure, so we do live imaging. We take an animal, we anesthetize the animal, and we use this toxin to visualize that same structure in the live animal, and then put it through some of these regimens, exercise, caloric restriction, or pharmacological intervention, and see how the synapse changes with time. So we do that, and when we do that, we have to be very careful that we don't use saturated levels of the toxin, because it will kill the muscle, or if it doesn't kill it, it will have a lot of adverse effects. And so we do have to be careful. In general, toxins do cause what they're supposed to do, what they do, which is death or abnormal changes.
Yeah. And then the last thing, I really liked your talk because I'm part Dominican and I'm from New York. So
I sense some bias here.
And you went to Keystone. Right?
And I went to the Keystone. And I'm also hitting that postdoc point. So knowing that when you made the decision after your postdoc to go to VTCRI, which was a new institute, like what drew you to a new institute versus an established institute?
Yeah, great question. So I have to be careful because I don't want to sound like too full of myself here. So I did have other options.
Who led up to McNabb lead?
I did have other options, the NIH, St. Jude Children's Hospital and Duke and other places. So there's pluses and minuses to everything. Okay, certainly there are pluses and minuses to everything. And so this is where you have to dig in and consider all the different things that, that are part of your life, right? Your work, if you're going someplace because of work, obviously they have to have what you need in order to be successful, because otherwise you're going to sink, right? But, you also, at least in my case, I have a wife and 2 kids, and I have family in New York City and other places. I want my family to be happy. I want them to be able to enjoy the place. I also want family and friends to be able to visit. And so driving to Roanoke was, as it would have been to DC. DC is actually closer to New York City. Most of my family is in New York City, in Connecticut, and the area in New Jersey. So there's a lot of different factors. What attracted me specifically to VTCR, so you take away all the geographical part component, that was a part of it. The school system was a part of it because I have 2 kids. Great school system here versus other places. Atlanta was another place. But what attracted me specifically to the work itself was that science, like most things, keep evolving. Right? There's a lot of departments and places that have been in place, including Harvard, that have been in place for many years. Very well established. But with that, you pay a price. There are more established investigators, so there's a lot of little kingdoms, and so people tend to talk to each other a lot less. As a new investigator, and I saw that while I was there, as people tend to to kind of push you aside a lot more, because they just don't really have time to help you, nurture you through that. It's a very different process of running a lab and being a PI than actually being a postdoc. And so you don't have as much exposure to the day-to-day working of an institute or a department. There's a lot of things that you just don't get your hands in a more established place. But I would say that the biggest negative for me in most of the other places is the lack of collaboration. I wanted to be in a place where I had the resources, but I was going to be able to talk to anyone, speak similar language, and just go at it. Let's go and do it. And there's a lot of barriers when you have people who have been entrenched for many, many years. And I felt that this was a place that had the resources in place. VT had made a commitment, Carrillo had made a commitment to give this place a shot at being successful. And I guess I have a bit of an entrepreneurial side of me. I wanted to be part of that. I wanted to make it happen, and I think it's exciting. As a new investigator, you have about 6 years, to really make a name for yourself. And I felt that it's a lot easier to do that when I'm having fun, with the people around me than being in another place where you're again in your classical research status, which is full wall, isolated. Long answer, but that was the honest truth. Yep.
Like Frank, I have a comment and then a question. But my comment is, you've all heard that Ed is always giving me a hard time about trying to eliminate the lunch because of saving budgets, while I'm really trying to prevent you from aging.
Don't buy that.
The question is it a reversible process? That is, if you're beginning to see the degeneration, if you start doing exercise, calorie restriction, do you see it improve?
So yeah, so with exercise, and so we did some live imaging of some of these animals. We did some live. So what I show you for exercise, was an already old animal. Okay. So this here is a 22-month-old animal. So presumably most of their synapses are like this. They have degenerated. They have changed abnormally. And then from another animal the same age, but that was allowed to exercise, the synapses look rejuvenated. Now these are 2 different animals, and so we still have the question, is it really that you're slowing further aging, or you're actually causing reversal of some of these abnormal topological changes that have happened? And so we took large number of animals, put them on the anesthesia, image them, put them back in a running wheel, in a cage with a running wheel, and then went back and looked at their synapses. So I took that off from this presentation because it would have been too much to explain, but in essence, you see reversal. So it is both. It is a slowdown of further aging, but it's also a reversal of some of the defects that accumulate.
I have a question. So you said, it's caloric. Is it the really calories, or is it the fat intake, or protein intake? So what exactly?
Good question. And so people are trying. People are testing a variety of different mixes, dietary mixes, to see exactly what it is. And so certainly we know the number of calories. If you exceed the number of calories, it's a big part of it is going to be fat, and that's going to lead to immobility or lack of mobility. Right? People tend to spend more time in their couches. So for this, that's I think the extent of it, is that the muscles and neurons are metabolically in tune much better because they're consuming exactly what they need rather than any more. Yep.
I was just wondering, so these images, so you mentioned that you had done this with a large number of animals. I was wondering about how much variation do you see with these different groups?
Yeah, excellent question. You see quite a bit of variation, but not as much as you'll be, as you may think. So, and you have to look at the animal as a whole. So some of these animals, because of aging, they will acquire cancer. They will acquire, their liver clearly is failing. So we take those animals out because they're not representative of normal aging. Those are animals that have had a disease for some X amount of their life. And so we take those. And so when you look at those animals, there is a lot of variability. When you look at animals that, as far as we can tell, looking at pretty much most organs and looking at their physical activity, just doing some general health assessment, there's very little variability. Yes?
I was looking at your, your first slide, so I was like, that was very impressive, your number of publications in graduate school. So you have any tips? I don't know how to do my research for me. But, I just saw 7, I was like, that's great. I don't know if they were all first author or co-authors, just a total number. But any tips just to get the writing done fast, or did you just kind of do everything first and kind of your last year?
Focus. I would say, when you have something that you think is going to lead to a publication, go after it. Put it, if you need to put it, if you have a side project, something else, put it aside. Focus on that main project, because that's going to give you a publication, unless you have time to really multitask. But so I had a couple of publications when I was an undergraduate student at Lehman College, and I learned that you have to manage your time very, very carefully, and ultimately, it's better to have one publication. We all dream about having, if you have a couple of projects that are going really well, having multiple publications within the same year. But with classes, with all the other stuff that is pulling us, it's hard to do that. And so the most important thing then is to get something out of it, and that is a publication. If you have something that you can write up, start writing it up, and as you start to put it together, you realize that there are holes. In your story, there's quite a bit of data that is missing, and then that's when you begin to churn, and you dive in and get everything that you need to complete the story, complete the publication. And it adds up pretty quickly when you do that. It adds up, and you get kind of more experience doing that, and eventually it becomes a little bit easier to prioritize your experiments and the writing and everything else. So.
Did you mostly collaborate with people in your lab? And so a lot of these publications, I know that in my lab we traditionally sort of work within our own lab, and we're working now with different collaborations, but were your advisors specifically, if you started a collaboration with another lab, were they really encouraging of that, or how did you navigate that process of?
Yeah, so I guess it's just part of me. My niece, one time kind of set me straight, because I was trying to act all cool and just Uncle, you're a nerd. This is like a kid from New York City growing up. Child, I'll get to you in a second, to your question. But, she sent me straight saying, you're a nerd, But I don't think that I'm your prototypical nerd. I think that, I like people. I like to work with people because I know that I have learned from so many different people. I've learned a lot we did 3 different science projects with my niece, and I learned a lot from her. It's amazing how someone who's naive to something that you're doing can ask incredible questions and just make you churn very quickly. So it's just part of my personality. I can't work in a place where it's very difficult for me where I can't be open and collaborative. Obviously, you got to be careful how much information you disseminate. There's certain things, it's not completely rosy, right? But it's part of my personality to seek collaboration. So when I was a graduate student, I collaborated with people at Hopkins, for example. I spent the big lab, my lab, Howard Hughes lab at Hopkins. I spent at least 2 months through the start of my graduate career working with people in that lab, and that led to multiple publications, a Cell paper, a JM Neuroscience paper where I was in the paper, they were in my paper. I helped them move their project faster, they helped me move my project faster. We accomplished our goals a lot faster. When I was a postdoc, the same thing. I collaborated with people throughout. Certainly my PI had a role in it, but as I got more seasoned, I started to have and establish collaborations on my own. And I would say if one thing that has helped me quite a bit, especially in my latter years as a postdoc, is the number of collaborations that I have had. Again, science, like most things, is very complicated. It requires a lot of resources and a lot of different skills and knowledge. And so you're not going to find that in your lab. I was in a big lab. I was in a lab of 22 people. Jason combined with another 144 people at Harvard. And we were still pretty limited, especially for a lot of the things that I needed to learn, and I wanted to just bounce ideas off from people. So I was in the main campus, by the way, not in medical school. So I established a lot of collaborations with people at the medical school. I established collaboration with people at MIT, established collaboration with people at Tufts, established collaboration with people in companies, Novartis and other places, because it helped. You got, as long as you're not, as long as it helps you, right? That someone, as long as someone doesn't steal your idea, you gotta recognize who you're going to collaborate with and who you're going to work with. But as long as you're not in any danger, right, of losing so much work that you put into a project, then I think it's more fun and it helps. So.
Okay, we got to bring Dr. Valdez back.